Okay, good morning, and welcome to this morning's session, which is the Neyman Award and Lecture. And my name is not Clifford Lam, my name is Piotr Rizovich, and it's my great pleasure to present this, chair this award. This year's recipient of the award in honor of a fellow Pole, Jerzy Neyman, is Professor Heping Zhang from Yale University. The Neyman Award and Lecture emphasize the interactions medicine. He directs the Collaborative Center for Statistics in Science, which coordinates the Reproductive Medicine Network to evaluate treatment mechanisms for human. He's a fellow of the American Association and a fellow of the IMS. He was the 2008 Mental Guru Distinguished Lecturer at the Harvard School of Public Health and an endowment lecturer here at the IMS. In 2011, he received the Royal International Award for Reproductive Health and the number of the president of the International and he's recently completed as editor of the JAMSA applications and case studies. And it is my great pleasure to present this award to Professor Zhang. I understand that there'll be a photograph taken. Excellent. Yes, Professor Zhang told me it's on Thank you for your kind introduction.
Good morning, everyone. I'm actually very humbled and grateful, and obviously honored to give this Neyman Lecture. I really want to thank the IMS for supporting me, and especially the IMS Special Lecture Committee for selecting me to give this lecture. It's really truly an honor for me. While I was preparing this lecture, I discovered I'm weakly related to Professor Neyman in a certain way. In 1984, I began to study statistics under the supervision of Professor Heping Zhang in China. And he happened to study statistics under the supervision of Professor Paulo Xu. And some of you may know, according to the information I found, Paulo Xu may be the first student of Professor Neyman, but it's a long time ago. So anyway, I guess if we try hard enough, you can always find some relationship among each other. Anyway, today I'd like to present what we have done in the past several decades in our understanding of this triangle relationship between genes, brain, and our health. And I should acknowledge this will be a summary of the work by my current students and former students. And I acknowledge some of them at the end. So here is the outline of my talk. I will begin with introduction, especially the data sources, and then I'll give you highlight a few challenges as motivation for our work. And for that triangle relationship, I will proceed in 3 steps. The first one is discuss genes and us. And I'd like to introduce a key concept that we have. It's called the supervariance. And I will get on to this brain and us. And the key idea we have is, so it's not our ideas, but the work we have done is related to tensor regression. And the last part is genes and brain. And that part related to analysis of data objects in metric space. For some of you who actually happen to join the research lecture by Professor Hans Muller, and his topic was on also on metric space. I was nervous when I heard his talk for the first time, but I think we have quite a different approach, and I hope to touch on why we use a different approach. And it's too early to say which one is better, but I hope we give you a different perspective. I will conclude with a few remarks and acknowledgement of the contribution that made this whole work possible. The Human Genome Project is really a landmark of scientific exploration in history. It began in October 1990 and completed about 85% in April 2003. Now, it's not entirely 100%, but it's close enough to be 100%. So what impact does Human Genome Project have in terms of scientific advance? It thirdly led to an explosive amount of data and information. That including here, UK Biobank is really, really wonderful resources. And in the US, we have PBGAT, and we also have European Commission's Human Brain Project. And the US also has a brain initiative, including Human Connectome Project, which I will revisit later. Now, what else? We know now pharmacogenomics has identified genes for which specific variants cause humans to metabolize drugs ineffectively, either too fast or too slow. Many of the causal genes have actionable gene variants. So actionable is actually very important. So whose consequences can be avoided by available medical strategies if we know what kind of genes we have. This includes cancer-driving mutations in tumors, and once identified, which can be counteracted by treatments with currently available drugs. So that's actually pretty important. And we have systematic approaches that improve our ways to diagnose cancer or hepatitis and other disease. And this can lead to evolution in terms of how we diagnose disease that will allow us to have early detection and follow treatment progression and treatment response to the treatments, and improving our ability to treat patients based on the medication we have now. So it does lead to important development. And most importantly, drug with support from human genetic studies for related effects that are reported in GWAS or single gene disorders databases succeed from phase 1 trials to final approval twice as often as those without evidence. So for pharmaceutical company, this is actually pretty important. So that's both time-saving and life-saving accomplishment. So what kind of challenge we face when we try to use and understand this kind of data? Most of the identified variants have a low associated risk and account for little heritability and creating so-called geneticist nightmare. And so that is, we know genes exist, we know they have effect, but we just don't know where they are, how they act. And one effective strategy is to aggregate thousands of minuscule effects, such as accounting for less than 0.05% of the trait variants through a so-called genome-wide polygenetic scores. And that's like our factor analysis. And a few such scores are found to predict, collectively speaking, about 10%, for example, for the variance in intelligence. And based on the classical study, especially twin study, we know 50% of human intelligence is attributable to genetics. So we now probably have sort of 10% identified. So 10% is still far away from 50%, but that's still a lot more than 1% and otherwise. So nonetheless, the missing heritability of intelligence remains substantial. We are talking about from 10 to 40 or 50%. And this is also similar to other complex disease. So a lot of work remains for us to do in terms of developing new methods and how we understand the gene works and relate to our human conditions. So we need to have improved analytic techniques to identify biological information in genomes, both functionally and evolutionarily. We need to rapidly analyze complete human genomes to identify and utilize actionable genes variants. And we need actually to better predict structures of biological networks directed from information of cognate genomes. So this means we have developed statistical methods that allow us to deal with high-dimensional and complicated data objects that include data objects in metric space. So now this section, I really like this because not only that we try to solve biological problems that let us challenges in statistical development. So the really ideal situation that we solve practical and scientific problems in the same time we advance our statistical science. Next, I will actually begin to discuss what we have done in this triangle relationship. And the first relation, we try to understand the genes on us. And as I mentioned earlier, we're trying to introduce this so-called supervariance concept. And so supervariants are set with minor alleles of single nucleotide polymorphisms at any loci of genomes. So we know genes usually sort of physically locate together similarly in similar regions, but actually for supervariants, it can be anywhere in the genome, just a sort of combination of anything. But for practical purpose, in statistics, in order to identify those supervariants, we generally speaking, we actually try to assume that relate to next to each other just for convenience. But conceptually speaking, supervariants are just combination of alleles of any loci. So if we put all the chromosomes together, this is not just one chromosome. You can imagine that we have all the human chromosomes put together. And as first step, we try to define blocks of chromosomes regions. And specifically, we consider one megabyte base pair for one region. And those regions can be overlapping with each other, but for convenience, again, we don't have sort of destroying regions of chromosome regions over here. And within each set of single nucleotide polymorphism, that I say SNPs, then we try to define the importance of each SNP before we construct supervariants. That reflect how the SNP actually will contribute to the trait that we're interested in. So in terms of a general framework of statistics, you imagine SNPs are our predictors, the axis in our regression model. And then the trait or phenotype as speaking is really the Y response. So for those who are used to imagine regression models, then the trait is really response or phenotype is a response, and the SNP is the axis that we are interested in. So the marginal importance of a SNP or predictor can be inferred from, say, univariate analysis, univariate regression, logistic regression, and linear regression. Now, if we try to account for both individual effects and hidden interactions, now, I will come back to these hidden interactions because that's actually a very important topic. Then we can use either random forest approach or even deep learning approach to sort of accounting for the importance of the contribution of each SNP. And so for forest, if we use forest, it always began with a tree. Okay? And so in the tree, we have given individuals with different traits or different characteristics of response. And then here, the predictors were mostly the single nucleotide polymorphism SNPs. And that's the X that we have. And we're trying to identify each SNP and then the minor alleles and the importance of a SNP based on where they show up in the tree. Okay, so this is not the Brayman's importance. This is our what we call a depth importance, where it depends on where you show up in the tree. And it's like an IMS. If you're really the president or the current president or the past president, you have more importance than me as a ordinary member. Okay, so that's how that we define the importance. So once you define the importance in the tree, then that will count in the forest. and then we take the average importance in the forest as importance for a particular SNP. Now, why we say it's reflecting both individual effect and hidden interaction, because your importance here and your importance here will be counted as individual and also interaction with the other SNP. Okay. That's why it allows us the ability to account for inactive effects. So this is how the process work in terms of how we use forest or even deep learning approach to assess the importance of SNP. And so we are giving a GWAS data set, and it could be another data set in your setting, and then we do a process of those data, either for quality control or for feature selection. I will describe what a feature selection is here. And once you have the process data, then we're going to use bootstrap to sample data 1, data 2, and data 3, and so on and so forth. And then for each data set, we're going to construct a tree model, and all those tree models together form this forest. And once we have this forest, then we're going to identify, we're trying to assess the importance of each SNP, and all those trees will contribute to the assessment of the SNP in this process. For some of you who actually, if you pay attention to this diagram, this is actually really similar to deep learning process. So if you forget about the contents inside, if you actually don't see the text, the picture is already almost identical to deep learning. That's why I really like to talk about the forest, because these days everybody talk about deep learning, so I have to make this connection. And we have this process described in our 2007 paper, that's a long time ago, published in PNAS. As some of you may know, the deep learning term was coined by Hilton in 2006. So at that time, I have no idea this is connected in any way to deep learning. But afterwards, when it became so popular, I said, like, my God, actually, it's similar to the ideas that we have, but we didn't have that term at that time. But nonetheless, you can see a lot of ideas come together in some way. So, and again, we try to use this forced approach to allow us to define importance for each SNP. And then these colors reflecting the importance of any SNP in a set. Now, if we back to statistical framework, you can imagine we have many predictors, tens or thousands or even millions of predictors, and that together with a set. And this whole process allows us to define the importance of each of those predictors. And why you think, well, why do we need to care about the importance of the predictors? Because not all the models are linear models. And so if we actually knows the importance of different predictors, that allow us to understand the models and predict in a different perspective. Now, once we have the importance defined, then we can decide a cutoff for based on importance. And how we define the cutoff, that is, so we have different individuals have different characteristics we're interested in, and so we're trying to cut off based on the importance scores. And the idea is to decide this cutoff is so that we have the greatest discriminating power against the trait or response that we're interested in. Okay. Once we have this cutoff defined, then we will be able to use that information to define what we call the supervariance in the end. So I'd like to note first, so the identification of supervariant was based on a discovery of the data, and then the estimation will be based on the validation of the data. Now, we can afford these days because, as I mentioned very earlier, that we have a database like Yuga Biobank that have 500,000 individuals, or other data sets have similar size. So we can afford, we don't have to worry about the bias estimation, so we can actually use discovery half the data for discovery, and then the other half of the data for validation. And we also usually repeat the same entire process again and again, for example, 10 times, and to make sure that the variance we identified will appear multiple times. And the reason we do so is to enhance the reproducibility of the discovery. And these days, one of the really big headache is really we identify some interesting result, and then when we try to do it again, it just disappears. So in order to enhance the reproducibility, we take extra cautions for our discoveries. Now here is mathematically how we do the feature selection in this kind of process. So as I mentioned earlier, we have SNP set, say G is a SNP set, and this V vector denotes the SNP vectors we have. And one of the key steps is really feature selection. That's again similar to the ideas in deep learning. When you get image data, you don't just analyze the raw image. You have to do a lot of processing. This is the second step, and you have to count for different potential relation. So here, if you look at this green and yellow tips, okay, and they're each reflecting a SNP. Say I have a SNP of 10. Okay, so I have 10 here, and the green says the major alleles, and the yellow are the two minor alleles. And so this vertical bar reflecting the importance of each of those 10 SNPs, and the rank like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And again, we're trying to find this minor alleles here. This is rank number 6 and number 8. So if we are given this entire information for the 10 SNP, but we define a new predictor based on the highest ranked yellow, that's 6. So when we, and the reason we have to do that, because number one, we have to reduce the dimension of the data. That's number one, step one. So we went from 10 variables to 1 variable. And number two, we have to reflect the information that we're interested in. So here is basically the X reflecting the first time a minor allele occurred for the particular individuals. And that's what we're trying to use to identify the supervariance. And numeric speaking, this is an example when we use supervariance to understand COVID-19 related mortalities. And we identify one region with 3 SNPs. And on chromosome 2, these are the alternation from major alleles to minor alleles. There's the frequency of the minor alleles. And when we say minor alleles or major alleles, the dependent frequency in the population, whether it's greater than 50% or less than 50%. So if it's greater than 50%, then we have major allele. If it's less than 50%, it's minor allele. We don't have 2 alleles. That's why it will be either 50 or less or greater. And scientifically, it's also interesting in the sense, if you look at the marginal odds ratio of each SNP, it will be between 1.14 and 1.33. That's why I said earlier, if you look at the individual SNP contribution, they're minuscule, they were small. But if you look at the odds ratio of the supervariance, it's close to 3, it's 2.7 something. And if you look at the marginal p-values of each SNP, that will be between 0.008 and 0.27, and that's not even near to any significance in terms of genome-wide association study. But if you look at the marginal p-value, if you look at the p-value of supervariance, that is 5 times 10 to the minus 8. So there is a great improvement of in terms of the size of the odds ratio and the level of significance. And of course, if you believe this result, that's actually major sort of shifting in terms of the size and magnitude and significance. And clinically speaking, we know one of the SNP is located in downstream of this gene TES and upstream of the gene SPEG. And they both report to be associated with what is called a cardiomyopathy, and cardiomyopathy was found to be related to the complexity and treatment for COVID-19. And so in a way, we found something, if that can be replicated, that has both specific implication in how we use information in a complex relationship, and also biologically, you may have a result that actually interpreted. Okay, so that's the part number one. I hope I explained to you how we construct supervariance and the potential for the use of supervariance. The second step one is related to understanding brain and traits. So when we say brain is reflecting mostly from fMRI data, and we call cancer because it has more than 2 dimensions. So we have fMRI data, and then we try to understand how we give the brain regions in the brain relate to cognitive score, say intelligence for us. Okay, so that's second relationship. And we may have additional covariates that including demographic information or genetic variables that we have just described. And in this model setting up, what we have, if we have response Y, In this case, could be intelligence score. And we have a covariance Z, and then we have this tensor, this fancy X is the imaging data, and this fancy B is the tensor coefficient associated with the tensor. So if you ignore this tensor part, then this model is no different from the typical ordinary linear regression model we have. We also consider a quantile regression model, and I will explain why we're interested in quantile regression here. And then in that case, we express quantiles in terms of covariance and then the tensor part. Now, again, if you ignore the tensor part, and what we have is no different from what we have done in the typical standard statistical regression analysis. Nonetheless, we cannot underestimate the challenges due to the presence of tensor in the model. The first problem we do is we have too many parameters in the tensor. So if you have D modes and each has a dimension P1, P2, and PD, that's the number of parameters with the product of those D numbers. And if you have 3, D equals 3, and each has 10, that's like 1,000. Okay, so it will be too many parameters we have to deal with. And one technique, well, not just one technique, basically everybody uses a similar technique, is trying to express a general tensor in a reduced but a systematic way. And one of the approaches is called the CD canonical polyadic decomposition. That's trying to express this tensor in a simple, mathematically simple form of shape. I will explain more how it looks like. But the key idea is trying to reduce the number of parameters from the product of several numbers to the summation of several numbers. Okay, so that's really the key ideas. And here's a reference that if you're interested in the details of this decomposition. Now, so here is the picture I really like to explain this idea. Okay. So imagine this room. This is actually much better knows. This is much better than the room I have across the street. Okay. So this you can imagine is a room here, and you can see here most of the space are empty, have no information whatsoever. It doesn't help you anyway. But after the room is consists of a lot of furniture. Okay. So in a way, it's really a summation of the information. That's number one. And number two, if you look at every these chairs or tables, they can be decomposed into simple geometric objects. If you look at the back of a chair, it's really like a flat block of the hand. Okay, so it's more almost 2 dimensions, and it is supported by this stand. That's really the one vector. So if you look at this, otherwise a nice looking complex 3D structure is really a summation of piece of simple structures. And that's actually essential. So that's how we use it. If you have 3-dimensional tensor, we're trying to decompose it in summation of simple structures. And each one of them is the name is chicken feet, chicken foot. And the next one I will show you specifically how we go from a simple structure to 3D tensor, okay, so that we know from one way to the other. And so we have A, B, and C, 3 vectors, 1 dimension, nothing fancy. And these 3 vectors, just for simplicity, it goes from 0 to 1 and to 0. 0, 1, 0, 0, 1, 0. Okay, so 3 vectors. And if you combine it together, you get a little cube in a space. And then if you add another 3 vectors, and then have another cube here, if another value, another cube here. So that actually you can fill the space with more objects. But each one of them were made up by simple structures. And that's important because that's how we go from the product of several dimensions to the sum of several dimensions, so that we can do some analysis. Now, after I don't have time to go through all the details. If you're interested, I'd be happy to give the specific reference. But just trust me, of all kind of manipulation, say we're interested in quantile regression of all those manipulations. At the end, we have to try to use what is known to us is basically optimization of some loss function plus penalizing. And these days you cannot get away without penalized regression. So why we have to use regularized or penalized quantile regression? Because even though we were able to reduce the dimension of the parameters substantially, but it's still a huge number of parameters we have to deal with. And that's on one hand. On the other hand, we're dealing with imaging data. We have to make use the spatial information in the data. And that actually unfolds case-by-case situation. If you have one kind of image, you have to use one sort of recorded penalty, and if you have another kind of imaging data, you have to use something different. Okay? So, but in general, this is basically the framework we have to deal with in terms of optimization. So we have loss function, we have penalizing parameters, and if you have that setting, then you can use something called the alternating direction method of multipliers for optimization. And that's what we did. So what I'd like to show you, one more step. When we have the dense data, we don't really know how many things we have to add together as a pieces. That's really related to in a matrix, but more or less like the rank time, the R. And we also don't know the tuning parameter lambda, like what we do in the penalized regression. So we have to use something called a Schwartz type information criterion, or you can also use generalized approximation consolidation procedures. But in both situations, we have something similar to the rank, that rank R. And that is what we call a Dix-Freeland in any model we have. Now, unlike the typical regression we have, even the definition of the Dix-Freeland for a model is not that simple or not uniformly agreed with. So what we have here as practical step, we just count how many non-zero elements in the penalizing term. There may not be, we don't actually know why it works in theory yet, and that's something that we're trying to study, but it appears to work in practice. And that's all we know for at the moment. So I will describe what we've discovered when we use this approach in an analysis of human connectome data. This is actually a large-scale project led by investigators in Washington University, University of Minnesota. And you had imaging data, we have behavior data, we have genetic data, we have a cognitive assessment. And the objective was to understand the brain function in healthy adults and understand the diversity of human behaviors. And you have 1,200 healthy individuals aged from 22 to 35 years old. Just 10 minutes? Okay, so try to catch up a little so to give you some potential question. So we have imaging as an access tensor predictor, and then we have this full intelligence score, Y as response. So first, how we define the response was basically you're given the squares and arrangement, and one is missing, and you have to say which one is missing. That's basically how we get the score. And the score goes from 0 to 27. And these are the information about the data I'm going to skip. And just go straight to the result we have. So these is the three direction how we see the brain is up, forward, and on the side. And so the color, the green indicate the regions most associated with 25% quantile, and the red indicating the regions that come from the medians, and the blue come from the 75% quantile. So number one, you can see that different quantiles to give you different regions. And this is why I said earlier why we do the quantile regression, because if you don't do the quantile regression, you don't see these different regions. And the other thing you can see that they are not symmetric in terms of hemisphere. Okay. So all the regions that we identified here are in the gray matters. The consistent brain regions relate to sensory perception, memory, and decision making. And so for the 25% quantile region, they're from the frontal lobes. The people know that actually highly relate to general and fluent intelligence. And the median quantile is the insular cortex that is known to be significantly related to adult intelligence. And then the 25% quantile is in the anterior singular cortex that's also attributed to high-level functions such as anticipation of tasks, detection, attention, and modulation or emotional responses. So even though theoretically speaking, we don't really know fully understand why we do this. So we sort of intuitive and reasonable, but actually when we apply this kind of model in the end, we were able to identify meaningful and interesting interpretive results. But it's left to us who are mathematical gifted and with a lot of statistical intuition, not only use this kind of model, actually trying to understand why it works. And this is why I'm actually really appreciate this opportunity to present this result to you and so to encourage you as, to actually study this kind of model and see why it has mathematically sound foundation. Okay, that's pretty much. So finally, I will discuss the last step is how do we view brain as an object. And Professor Hans Sumula give you enough motivation why it's important and interesting to study an objective metric space as an interaction. But nonetheless, geometric data these days are really common, and you will see, including facial recognition. And they actually do not fall in the typical Euclidean space that we usually deal with, and sometimes fall in the so-called metric space. I learned metric space a long time ago. When I worked with my student, he kept reminding me what the heck is metric space. So the metric space is a non-empty set together with a metric on the set. It doesn't have too much information. And the metric measures the distance between any 2 points on the set. So we basically have a bunch of sets, and then you just have distance among any two. And that's basically the information we have. Now, in the Euclidean space, we have a norm. The norm can be used to define the distance. So the Euclidean space is a special situation of metric space. And so for statistics, Chris yesterday gave his philosophy on probability and statistics. So what I see is we're trying to understand uncertainty, and that's measured by probability measures. And we use data to understand the uncertainty. But in my humble understanding, it's usually not that easy to understand directly the probability of uncertainty, and we go to the so-called distribution function. And then we use data to measure the estimated distribution function, and then we have this Korilenko-Canteri properties and thanks to the theory to allow us to be able to say, okay, I feel pretty good about using empirical distribution to estimate the underlying distribution. And then we use this correspondence theory to say, okay, I understand the distribution function because the one-to-one correspondence between distribution and the measure, I know what happens to the underlying mechanism to generate our data. That's actually what we have in the Euclidean space. But what happened in the metric space and what we have to do? Now we know that whether you are in the metric space or whether you're in the Euclidean space, you can always have uncertainty. So this is metrical theoretical foundation for probability. That's not an issue. And then you can always generate the data because we see images, we see you and you see me, so data is not the issue. But what's missing? We need a distribution function. We need to be able to estimate the distribution function. We need to be able to establish the connection between probability and distribution. We have to ensure that empirical distribution will converge to distribution. As far as I know, we don't really have good answer. Okay? So that's actually the part I'm trying to work with my student. To be honest with you, is mostly my students' work. And so we are in the process of revising a paper for JASA. Now, don't ask me why we actually sent to JASA, not to the others, but nonetheless, that's a different story. So we actually establishing the one-to-one correspondence. We're trying to define how to estimate empirical distribution, and we're trying to actually establish a theory why the empirical distribution will converge to distribution. But the key step here is how do we define distribution function in metric space. Actually, that's not actually easy because in the Euclidean space, we have vectors, we have rectangular, and we define distribution in one direction. Okay? One direction. But when you deal with metric space, we don't have the one direction anymore. We use actually balls. It's a different topology. And the distribution we have in the metric space with the metric space distribution looks differently. Okay? That's a shape that we define a distribution function in the metric space. And so we took a different approach than the depth and what is called optimal transport function. This is a very popular technique. We use a different approach. I'm just explain how we approach it. So we imagine this axis. So those are points we observed from metric space. We take every point as a center, And we define the radius between this point and the other point as a radius, and define a sphere around that for these 2 points. And then that's 1 point and another point. We're going to define another sphere. And for this point, we're going to another sphere, and then this point, another sphere. So we have one center, and then mutually we define spheres. And then we do the same thing for each point that we observe. So we have many, many spheres. So it's like you walk on the earth at night and just look at the sky. Oh my God, it's beautiful. Okay, so that's how we. So this is us looking, walking on the sphere, the earth, and then this one star, one star, one star. Okay? And we use the measures of those spheres as a way to define the distribution function. Now, when we so this mathematically, I'm not, because of time, I'm not going to go through detail because you guys just see in a few seconds, how I define it. But the idea is speaking is a difference between the metric space distribution and typical distribution. In the typical distribution, we define in linear space is really a function of one vector. But here, a metric space distribution function is a function of 2 vectors. Okay, and one vector used to define the center, and the other vector is to define the distance. So because of that you really you have to redefine all the theories, but they all sort of connect through this something called a concentration function that is a measure probability in this neighborhood, and then allow us to establish the relation between the typical, the standard distribution function theory and then metric space distribution function. As I said earlier, I have to point out this work is mainly done by my former student, Heping Zhang, and then his students' work. And just before I conclude with this part, I would like to point out as the why we take a different approach, because one approach is use, as I said, gaps and then optimal transport. But that approach, to our understanding, it depends on some first level optimization in metric space. That can be challenging to begin with. So this is why we actually trying to use 2 vectors to define distribution function. As far as we know, we try to have an approach that robust to small sample size and also deal with presence of outliers, and also importantly that the approach we actually propose is really available not only for sort of one space, metric space, but it can be for joint metric space. Okay. So back to the triangle, a summary, what we have contributed to this understanding. Now I can introduce this concept of supervariance and for genomic data, because that's how I get paid for. But the same ideas can be used for non-parametric or non-regression. It's a particularly convenient approach to deal with hidden interaction. To me, that's actually pretty important and challenging topic. And I present you with this tensor regression model using quantum regression and regularization ideas. As I admit, you have something it seems to work, but we don't understand deeply how the theory behind it. And finally, I introduced this metric distribution function to conduct statistical inferences for data objects in metric space. And more to come. I hope I have more opportunity to present the result to you in the conference. And as I mentioned, nothing that I present here will be possible without the contribution of many of my students, including Xianbi and Xiangchen and Wei Dai and Longfeng and Jianchang Hu, Cai Li and Qing Li, Qi Song and Xu Wang, and most importantly, Heping Zhang and Hong Tu Chu. They are really sort of essential to my work. I'm grateful to the support of National Institute of Health and also National Science Foundation. And the data made available to us by Human Connectome Project and Yugo Biobank. And the Yale Center for Research Computing has been essential to our work. Thank you so much. In the matrix, the response is the brain. Okay, so I have a three part. The second part, the response is IQ, and the brain is predictor. Okay, in the last part, the brain is the response. Yeah, yeah, yeah. Yes. Yeah, that's a very important question. That's before I try to quantize, I thought, well, you know, you're my student, we have the preparation, just go ahead and try quantize. But when I see the result, I thought, now I'm convinced that the quantum regression is useful, because if you use different quantize, it will be different discoveries. You don't see things if you just run regression, linear regression based on the means. And if you use different quantize, and so this is for one quantile, this is the other quantile, and this is the other quantile. So you actually see different result. Now, what I don't know is whether this result, how replicable this result will be. But if it is true, then that's indicating the usefulness of quantile regression based on different quantiles. Yes. Thank you. That's a very interesting, important question. Now, so one thing we did is we tried to so we have importance based on the kind of model structure, but we also have importance based on what we learned from other literature, which are integrating information together. But I have to admit, we have not formally compared the importance ranking from one way to the other. But I think that's a useful thing to look into. Yes, yeah. Thank you.